Imagine for a second shrinking down and literally walking through the stromal extracellular matrix of a tumor.
Oh, that's quite a visual.
Right, like you're navigating this crazy cellular metropolis, and you're not just, cataloging what cells are there, but you're looking at the literal microscopic zoning laws.
The structural strategy of it all.
Exactly. Like who is physically touching who, where are the barricades, how does modeling the local architecture to build a heavily fortified outpost in your lymph nodes.
It's a completely different way of viewing oncology.
It really is. So, okay, let's unpack this. The mission for today's deep dive is to figure out exactly how one of the most aggressive cancers out there, small cell lung cancer or SCLC, spreads and how it outsmarts the immune system.
And SCLC is notorious, right? It makes up about 15% of all lung cancers, and it metastasizes incredibly fast.
Fast and aggressive. It goes to the lymph nodes, the brain, the liver, the bones.
Yeah.
And to understand how it does this, we're pulling from a groundbreaking 2026 paper from Cell Reports Medicine.
Yeah, and the reason this paper is so revolutionary is because of the tech they used. It's called single-cell spatial transcriptomics, specifically the CosMx SMI platform.
Which sounds like a mouthful, but I love the analogy you used before we started recording, the smoothie versus the map.
Right, yeah. So think about traditional bulk RNA sequencing. That's like putting a tumor in a blender and making a smoothie.
A really gross smoothie.
Very gross. But, you get the smoothie and you know exactly what ingredients went into it. You know there are 50% tumor cells, 30% T cells, 20% fibroblasts.
But you have zero idea how they were arranged before you blended them.
Exactly. You destroy the tissue architecture. You don't know if those T cells were actually fighting the tumor or if they were like miles away on the periphery.
So spatial transcriptomics is the alternative.
Yes. It's like having a high-resolution Google map of a city. The CosMx platform uses these in situ hybridization probes to tag RNA directly on an intact slide.
So you see where everyone lives.
You see where every single cell lives, who its neighbors are, and who it's actively avoiding.
And the scale of this data is just massive. They mapped over 600,000 individual cells.
Across 105 samples from 75 patients.
Which is wild. And they divided these patients into 3 distinct groups, right? Which kind of gives us this evolutionary time lapse of the cancer.
Right. So you have the PT group, which is the primary tumor without any lymph node metastasis.
The cancer that stayed put.
Exactly. Then the PT-LNM group, which is the primary tumor that has spread to the lymph nodes.
Okay, the mobilized one.
And finally, the LNMT, which is the lymph node metastasized tumor itself, the actual settlement.
So we have our map. We have our timeline. Who are the bad guys driving this spread?
Well, out of the 13 distinct malignant subclusters they found, 3 of them stood out. C5, C6, and C9.
The vanguard.
The metastatic vanguard. They were almost exclusively found in the tumors that spread the PT-LNM and LNMT groups.
Okay, I want to dive deep into these 3 because they don't just divide. They, engineer their surroundings. Let's start with C6. I like to think of C6 as a stealth bomber.
That's a great way to put it. C6 is all about metabolic reprogramming and immunosuppression.
Right. It heavily expresses 2 genes in particular, SERPINA1 and DHRS2. So how do these act as the radar jamming chaff and the alternate fuel source?
Well, SERPINA1 is a fascinating defense mechanism. It codes for a protease inhibitor.
Meaning it stops enzymes from breaking things down.
Exactly. In a tumor, immune cells secrete these proteases, basically chemical rounds, designed to break down cell membranes so they can reach the tumor.
Okay.
By upregulating SERPINA1, the C6 cells secrete their own inhibitors to neutralize those destructive enzymes before they even hit.
Wow. So they're literally coating themselves in biological Kevlar.
Yep. They neutralize the immune attacks in the extracellular space. And then you have DHRS2, which shifts lipid metabolism.
The alternate fuel.
Right. When a tumor cell tries to settle in a new niche, it's usually starved of nutrients, especially lipids, which it needs to build new membranes. DHRS2 lets C6 alter its pathways to survive in that desert.
Okay, so that's the stealth bomber. Then we have subcluster C5, which expresses FYN and NR2F2. These are all about movement, right?
Yes. FYN is an enzyme that regulates how a cell rebuilds its internal skeleton.
Because to migrate, a cell has to literally dismantle its own scaffolding and rebuild it.
Exactly. Actin filaments. FYN hyperactivates that process, giving the cell the mechanical flexibility to squeeze through blood vessel walls and navigate dense tissue.
And NR2F2.
It pairs with FYN by regulating genes for tissue invasion.
Okay, and the last of the vanguard is C9, relying on the HIF1A gene.
Hypoxia-inducible factor 1 alpha, which makes perfect mechanical sense.
Because as they pack into the lymph node, they run out of oxygen.
Right. The core becomes severely hypoxic. So HIF-1A activates this massive shift to anaerobic glycolysis. C9 survives where oxygen is basically zero.
Oh, wait, I have a major pushback question here. I'm stuck on the geography. Okay, lay it on me.
You're saying C5, C6, and C9 are thriving in the lymph node. But the lymph node is literally the headquarters of the immune system. It's a high-density garrison of immune cells.
It is.
So how does a cancer cell physically survive that? Does it chemically kill all the immune cells or just, like, shove them out of the way?
It shoves them. It's a phenomenon called immune exclusion.
A force field.
Basically, yes. The spatial map showed that even though there are tons of immune cells in the lymph node metastasis, these vanguard subclusters have a distinct physical buffer zone around them.
So they aren't necessarily killing the immune cells right next to them.
No, they use dense matrix deposition and contact-dependent repulsion to literally push the immune cells to the periphery. It's a sterile perimeter.
Okay, let's pause here for a quick mental recap for everyone listening because that was a lot of dense biology.
Good idea.
Number 1. SCLC spreads aggressively, and we are using spatial mapping to see where cells live, not just what they are.
Right.
Number 2, the cancer uses a vanguard subclusters C5, C6, and C9 to invade. And number 3, these vanguard cells survive by building a spatial force field that physically locks the immune system out.
Perfect. So moving on to segment 2, here's where it gets really interesting. If the vanguard is putting up force fields, what happens to the immune cells? They can't just be standing around.
No, they don't just disappear, but the composition of the immune response completely flips.
Right. In the primary tumors that haven't spread, the map shows tons of effector T cells.
The active soldiers fighting the tumor.
But in the lymph node metastasis, those soldiers are gone. The landscape is suddenly dominated by exhausted Tregs regulatory T cells and naive B cells.
It's a highly orchestrated shift. The researchers mapped this transcriptional continuum. They found 11 specific genes that steadily increase as the tumor moves from the primary site to the lymph node.
And these genes are involved in chemokine signaling and NF-kappa B pathways.
Exactly. The tumor is literally hijacking the immune system's communication lines.
It's setting down chemical breadcrumbs to recruit the Tregs.
Yes. The Tregs suppress the immune response. So the tumor is bringing in cells that will protect it while creating an environment that suffocates the active soldiers.
So the vanguard cells are acting like bouncers at a club.
That's a great analogy.
They're pushing the effector T cells out the door, but if the T cells are pushed out, where do they go? They have to pool somewhere on the biological sidewalk, right?
They do. And this is one of the most striking findings. They found the displaced immune cells pooling in the perivascular niche.
Around the blood vessels.
Yes. The endothelial cells lining the blood vessels actively avoid the malignant tumor cells. Instead, they form these tight physical networks with the trapped immune cells.
It's like the blood vessels are hugging the immune cells for safety at the edge of the tumor.
It's exactly like that. Vascular-immune crosstalk. And what's fascinating here is the tragedy of this arrangement.
The irony of it.
Yes. When you look at the RNA of those trapped T cells, they aren't exhausted.
They are expressing super high levels of GZMA and GZMB, granzyme A and B.
The cytotoxic weapons.
So these T cells are armed to the teeth.
Fully activated. But because of the spatial barricades, they are physically sequestered. They're trapped near the blood vessels, completely separated from the tumor cells they want to destroy.
Oh, man. So you have live artillery sitting just like blocks away from the target, fully operational, but structurally cut off from crossing the street.
The cancer defeated the logistics, not the hardware.
Okay, time for mini recap number 2.
Go for it.
Number 1, the immune response shifts from active soldiers to exhausted regulatory cells as the tumor spreads.
Number 2, the tumor achieves this by hijacking communication pathways like chemokines.
And number 3, the armed immune cells don't die. They are just physically trapped near the blood vessels, unable to reach the tumor.
Which perfectly transitions into the macroarchitecture, because we know where individual cells hide, but how does this entire city layout impact the actual patient in the hospital bed?
Right, because that's what really matters.
Exactly. So the researchers looked at cellular neighborhoods, or CNs.
Basically zoning districts.
Right. They evaluated the 10 nearest neighbors of every single cell, and they found 6 major spatial structures.
Things like the exhausted niche or the tumor compartment. But the protagonist neighborhood here, the one we really care about, is PIHS1.
pan-immune hotspot 1.
Such a catchy name.
Scientists love their acronyms, but PIHS1 is a highly coordinated, tightly packed pocket of interacting T cells and B cells.
It's the good neighborhood.
It is. It's a zone where immune cells are actively communicating and mounting an attack.
And the data shows that the abundance of PIHS1 is highest in the primary tumors, but the cancer systematically dismantles it. It almost entirely disappears in the lymph node metastases.
And if we connect this to the bigger picture, the clinical takeaway is massive. Patients who maintain a high abundance of PIHS1 have vastly superior overall survival.
The hazard ratio was, what, 4.418?
Yes, meaning if your tumor dismantled that specific PIHS1 neighborhood, your risk of mortality was more than 4 times higher.
That is staggering. But wait, I have to play devil's advocate here. If we just need to know if there are lots of T and B cells, why do we need $1 million spatial transcriptomics machine? Couldn't a doctor just run a standard biopsy and count the total number of immune cells?
It's a logical question, and the researchers checked that. They counted the total density of immune cells using bulk sequencing.
And
it had 0 predictive value for survival.
really, 0.
0. The total number didn't matter. You could have a tumor completely flooded with immune cells, but if they aren't physically arranged into that PIHS1 neighborhood, if they are just scattered or trapped near the blood vessels, they are functionally useless.
It really is all about location, location, location. Density doesn't equal coordination.
Exactly. The functional unit of immunity isn't the single cell, it's the neighborhood.
But that creates a huge problem for everyday clinics, right? Like, a community hospital doesn't have a CosmX machine.
Which is why the researchers did something brilliant. They translated this spatial map into a 20-gene signature.
Okay, explain that.
They found the top 20 genes uniquely upregulated within the PIHS1 neighborhood. They took that signature and applied it to standard, non-spatial biopsy data.
So even without the map, the strong presence of those 20 genes acts as a proxy for the neighborhood.
Right. It bridges the gap between high-tech spatial discovery and everyday clinical use. They proved you can predict survival using this signature on normal biopsies.
Okay, final mini recap. Number 1. Cells form specific functional neighborhoods based on their 10 nearest neighbors.
Number 2, PIHS1 is a highly effective protective neighborhood of T and B cells.
And number 3, the physical architecture of PIHS1 predicts patient survival way better than just counting the total number of immune cells.
Spot on.
So what does this all mean? Like, why should you, the listener, care about cellular zoning laws?
Because it completely changes how we think about fighting cancer.
Exactly. For decades, we've thought of metastasis as just unchecked cell division. And our solution has been to build better molecular assassins drugs that find a bad cell and kill it.
But this paper shows that the tumor is surviving through architectural engineering.
It's building force fields with C6 cells. It's trapping our armed immune cells near blood vessels. It's actively tearing down the good PIHS1 neighborhoods.
So throwing more assassins at a logistics problem won't work. If the resistance is spatial, the therapy must be spatial.
We need to degrade the barricades, drop the SERPINA1 shield, break down the extracellular matrix, and let the immune system, which is literally waiting right across the street in the perivascular niche, flood the tumor.
It's a paradigm shift.
It really is. And it leaves us with this final kind of provocative thought to chew on. We spend billions developing drugs to act as assassins. But knowing what we now know about cellular neighborhoods, what if the next generation of cancer treatments aren't assassins at all?
Right.
What if they act as urban planners? Molecules designed to actively rezone the cellular real estate, tear down the vanguard's barricades, and finally let the immune system cross the street and do its job.
It's an incredible way to look at it.
Think about that next time you look at a city map or, try to navigate a detour. Sometimes the biggest obstacle isn't the strength of the enemy. It's the layout of the battlefield.